Hi everyone, I'm Ömer Sümer and I will present our work, Region-Based Saliency Explanations on the Recognition of Facial Genetic Syndromes. Many rare genetic conditions cause facial dysmorphism and they can be difficult to recognize. Machine learning can help physicians' diagnosis and decision-making. Deep learning has also been used on this task, as in a recent work by Krawitz and friends, Phase 2 gene. However, DNNs lack internally interpretable mechanisms. They are trained with class labels. In contrast, physicians have years of clinical practice and knowledge such as phenotype ontologies. In this study, we propose an approach for region-based saliency explanations and how DNNs and physicians' decisions are in line with each other. Now let's have a look at our approach. We first train a DNN to classify different genetic conditions and unaffected group. Then we use a saliency method to explain trained DNN's decision and aggregate pixel-wise values to regions and investigate their contributions to the DNN classification. In this example, we see the region relevance values highlighted based on a given saliency map. Later, we will compare them with phenotypes observed by clinicians. Our dataset contains 3,547 images of 11 rare syndromes and unaffected individuals. All the images are acquired from scientific articles using Google Scholar and PubMed. Further, they are all verified by clinicians to prevent incorrect diagnosis. We use visible facial phenotypes from clinical synopsis section of OMIM as described in the human phenotype ontology. 2 to 3 clinicians viewed 171 images and labeled the presence of these phenotypes. We will need them to evaluate region-based explanations. We focus on 2 research questions. The first, do the region relevance coefficients enable us to distinguish syndromic and unaffected samples? The second, do region-based explanations align with HPO terms labeled by clinicians? Our approach is independent from any saliency methods. We use 15 different saliency configurations and compared them. Here we see the number of statistically significant tests for each region and syndromes. These results are in line with the most prominent facial phenotypes of 5 syndromes that we focused. In correlation analysis between region-based saliency coefficients and the amount of observed HPO terms by clinicians, mouth region is depicted well. Best performing deep-lived, GradCam, and occlusion-based methods. Similarly, guided GradCam and GradCam capture eye region. However, they mostly fail to capture the dysmorphism in the nose. 3D understanding may be required to depict short columella or bulbous nose. In conclusion, our study created an important link between DNN decisions and HPO taxonomy. In order to use saliency explanations in clinical decision support, they need to be more aligned to HPO ontology. We also aim to conduct a large user study and improve the phenotyping performance in a future work. Thank you very much for your attention.